Kolind completed her graduate studies originally in physics at the University of British Columbia under the supervision of Dr. Alex MacKay, where she worked on developing quantitative MRI methods for measuring myelin content. And then she took up MS Society of Canada Postdoctoral Fellowship, first at Oxford University and then at King's College in London, under the co-supervision of Dr. Deoni and Heidi Johansen-Berg, where she continued working on these methods as well as their various clinical applications. And after her time in the UK, she then returned back home to Vancouver, where she took up a Michael Smith Foundation postdoc, working again on these same methods, but more of a focus on MS, actually working with the director of the UBC MS Clinic and Research Center. And just for those of you who haven't met her and maybe not, might not be aware, Shannon's been heavily involved in the development of some of the methods that she's going to be talking about today, and has basically has authored or co-authored a number of seminal publications in this area. So we're very happy that she came all the way from Vancouver to tell us about her work.
Thank you for coming, and thanks so much, Chase, for hosting this visit. It's been great. Enjoying the blue skies and the break from the rain in Vancouver, and it's been great to meet so many people, so thanks for taking the time. So yeah, I'm going to talk about an MRI technique that is hopefully specific for myelin, and I'm going to talk you through a little bit of the development of it up to where we are right now, just starting to put it into clinical trials. So I understand that a lot of people don't have a background in MRI, so I'm going to start with an embarrassingly basic, hand-wavy, intuitive, hopefully, description of MRI and how it all works. And feel free to ask questions. So again, very basic. So most of our body is made up of water, and so we have these lovely two little hydrogen protons on the water molecule, and these act as little magnets. They have a magnetic moment, and when we have just some water, then they're all going to be oriented in a random direction, their magnetic moment. But if we apply big magnetic fields, then they'll line up either with or against the magnetic fields. And there's actually a very small proportion more that line up with the magnetic field than against it. So this gives us a net magnetization, so we can actually measure this amount of magnetization that is pointing along the direction of the external magnetic fields. So with MRI, we're usually looking at the effects of relaxation. So in the natural state, we have this big external field that we're applying, and we have your brain, which is essentially called a bucket of water. And we have this proportion of protons that are aligned with the magnetic field more. So we're measuring this net magnetization M naught, so we can measure that amount. Then we put some energy into the system and we flip them all by 180 degrees so that there's now a net magnetization against the external magnetic fields. So now we measure negative M naught. And if we just leave it alone for a little while, then they will start going back to the lower energy state. And this happens in an exponential fashion. And so we characterize the time that it takes for that to happen using T1. So T1 describes the time it takes for it to go back to its natural state. And so 63% of the signal is recovered at a time of T1. And T1 is going to be influenced by a lot of different things, but primarily by just free water content. So how much water is there, essentially, will dictate what your T1 time is. But it's also going to be affected by things like the presence of macromolecules and lipids and things like myelin, and motions and temperature and magnetic fields. So it's not simple, but overall it's telling us something about water. And then we can look at a different type of relaxation, so T2. So now I've flipped the axis, so we've now got the magnetic field coming out of the screen. And so we start out with our spins aligned in that direction. And instead of flipping them by 180 degrees, we just flip them by 90 degrees. So now our magnetization is pointing up. And if we're measuring the signal along the x-axis there, we start out with that signal. And on a much shorter time scale than the T1, they start to interact a little bit with each other. So they lose energy to each other and they get a little bit out of phase, out of sync. And so the net magnetization that we measure along that x-axis decreases, and that's also exponential. And we characterize that with T2. And so that just describes how long it takes basically for them to lose their coherence, so that we're not measuring that magnetization along that axis anymore. And so again, this is going to be influenced by how much water there is, but it's even more sort of influenced by what is around the water. So how tightly constricted is it? How much are they going to interact with the other water molecules? Is there something paramagnetic nearby that causes them to get out of phase more, like iron? So these are generally the two characteristics that we're looking at with MRIs, T1 and T2. So generally with MRI, we're doing qualitative imaging. So this is a T1 weighted image and a T2 weighted image from a healthy control. So basically we're taking these intrinsic properties, T1 and T2. We're not actually measuring them, but we're using those properties to weight the signal. And so the contrast is dependent on the T1 and T2 of the different tissues. And so you see brighter values for white matter or gray matter or lesions or CSF or different things depending on the T1 and T2 of the tissue. And so that's really useful. Obviously, this is the same slice. It's for the same person, just with slightly different scan parameters, and so you get a quite different contrast. And this has been huge for the study of MS, which is where I spend a lot of my time and research, because you can identify these lesions. So you can see these bright spots. Both of these patients have some bright spots. They're harder to see on the patient on the left. So this helps with diagnosis. But MS is actually a really complicated disease. So there's a lot of different pathological processes that are going on, possibly all at the same time. So you have inflammation and edema, you have gliosis, and then you have loss of axons and demyelination. Demyelination is really the hallmark of MS. And so one of the downsides of conventional MRI is that we have these lesions, but we don't know what is happening in them. It could be any or all of these pathologies. So we don't know if this is just simple inflammation or if there is actually demyelination and axonal loss in them. So that's lacking specificity. We're also lacking sensitivity. So we can see these lesions, but we also know from animal studies and from histopathological studies that these processes are also going on in the tissue that looks normal on these images. So we need to be more sensitive to those changes, not just seeing the lesions. And we also want to be more quantitative. So if we do the same image twice, so if I put the same person in the scanner and scan them twice, you can't just subtract the images from each other because the window and level is set differently every time. It's different on every scanner every day. So it's really hard to make quantitative comparisons. And anytime that there's an observation made, you can say something's bigger or smaller, brighter or darker, but it's going to be relatively subjective. So we want to get more quantitative so we can be more sensitive to small changes. And there's also an issue of clinical relevance, especially in MS. So these two patients actually have the same disability score. So they actually have very similar low disability. But probably everyone would agree that you would have guessed that the patient on the right had a worse disability if you see all these lesions, but actually they're pretty much the same. So just how many lesions or what volume of lesions you have isn't enough to predict how bad your disease is or when you're going to start getting worse or your prognosis. So this is the best picture I could find for non-conventional MRI. So it's a PASTA scanner that we did for us in Italy. So just terminology-wise, when I say conventional MRI, I mean things that are routinely used in clinic. Non-conventional or advanced MRI, I mean sort of everything else. So what we're really looking for in a non-conventional MRI technique is something that's more sensitive, so that we're picking up changes in the tissue that looks normal, and more reproducible, so that we can do quantitative comparisons between groups, but probably more importantly over time, so we really know what's changing in a particular patient, and more specific, so we'd like to know what it is that's changing. And in particular, we're interested in something that's specific to myelin because we're so MS focused at the moment, and that's kind of the hallmark of MS. So I've told you a little bit about relaxation. I'm going to tell you about quantitative relaxation imaging or myelin water imaging. Okay, so as we already went over, MRI is generally looking at signal from water, and we tend to assume that within 1 pixel of our MRI image, that water is in sort of a homogeneous environment. But of course, that's not true. If we look at a cross-section through this cartoon, myelinated axon, you see water in lots of different environments. And from a relaxation point of view, we can broadly separate this into 2 categories. So we have water that's in the intra- and extracellular spaces. And so because this is fairly unrestricted, the relaxation times are pretty long, so the T2 is longer. But we also have water that's trapped in between the bilayers of the myelin. And this is really restricted, and so it relaxes quickly, so it loses that coherence really quickly. So this is the key to the multi-component relaxation imaging, is that we separate out how much of the signal is coming from each of these 2 different water components, even within 1 voxel, okay, or 1 pixel. So essentially, I talked about the T2 looking like an exponential, so you excite the signal and you just watch it decay. It's a pretty simple experiment. And usually people just fit this to a single exponential and say that's your T2, or weight by it. But we just fit it to a multi-exponential, so it's actually the superposition of these multiple pools relaxing at different rates and in different proportions. So we just fit it to a multi-exponential, and we get out a T2 distribution. So this tells us how much of the signal within that 1 pixel has a particular relaxation time. And if you do that in brain or spinal cord, you generally see 2 peaks. And so that the longer peak corresponds to the intracellular water, and the shorter peak is from the myelin water. And we quantify this by just dividing the area under the myelin water peak by the total signal. We call this the myelin water fraction. So you go through and you do that at every pixel, and you get out a myelin water fraction map. So this is a quantitative MRI image, so it really is related to how much of our signal is coming from myelin. And it should be the same every time you do it, so it's an actual number, it's not just a weighted image. So it is actually related to myelin. So the next step was to try to do some validation. And so this is work done by Corey Lowell in Vancouver. And this is a post-mortem MS tissue. And so you can see a conventional image on the left, and then our myelin water fraction map, and then this is a stain for myelin lipid Luxofast blue. And so you can see a pretty good correspondence between the myelin water map and the Luxofast blue. So where it's bluer, there's higher signal on the myelin water map. And she went through and did that quantitatively on a bunch of different samples and found a strong correlation in each sample between our myelin water fraction and the optical density of the Luxofast blue, so how blue is the blue. And this sort of falls across all of these different types of tissue, so within white matter, within gray matter, and lesion, and so on. So that's really encouraging that what we're measuring is related to actual myelin content. And then there's also been some preclinical or animal models. So there's rat sciatic nerve on a tellurium diet, and they found a good correspondence between that myelin water fraction and this was EM measurements. And then spinal cord injury, that was again done at EBC, and a good correlation between the myelin water fraction maps and Luxofast blue. There's been a few other studies in rats primarily, guinea pig as well. And some of the things that we found out is that this myelin water fraction is sensitive to myelin debris. So when myelin has already been hurt, but it's still hanging around in the macrophages and so on, we're still picking up the signal from it. So we don't really know the time scale of that in MS, but we have to be aware that we're still picking up signal immediately after injury. And also, by the same token, we're not very sensitive to early demyelination, so we're not picking up this loss of myelin signal until it's really kind of gone. So just some of the caveats of the technique. All right, so then we got to starting to apply it in MS. And this is just an example on the bottom of a conventional image. And you can see the bright lesions. And then the second image is they have gadolinium, so that changes the relaxation time so that you, anywhere that there's a breakdown in the blood-brain barrier, so it becomes really bright wherever there's active changes in the lesion. So you can see one lesion there that's actively changing. And then the myelin water fraction map. And you can see the lesions showing up in the myelin water fraction map, although some are darker than others, so there is still some myelin remaining in some of the lesions. But you can also see the changes kind of more diffusely. So I put some red lines there to sort of hopefully guide your eye that the center of the image looks a little bit darker. So there is diffuse reductions in the myelin water fraction that you can't pick up in the conventional image. And so you can quantify that, so you can go through and actually measure, what is the average value in that normal appearing tissue. And we always come out about the same, about 15% reduced for MS patients. So we're able to pick up these changes not just in lesion, but in that normal appearing tissue. There's also sort of a gray area, so to speak, between lesion and normal appearing. So there's this diffusely abnormal white matter. And it turns out that the myelin water fraction is also significantly decreased there. And that's a really interesting area because nobody really knows what this represents, if this is sort of pre-lesional or if it's an indication that you might progress faster. So that's an area that we're looking into now. We're better at picking out differences between lesions. So like I said, we're not specific with the lesions, so we can now tell which lesions are really demyelinated and which ones still have a good chance of recovery and so on. We've been able to measure changes in the myelin water fraction in lesion over time, and an early study we were able to show changes in myelin water fraction in the spinal cord over 2 years in primary progressive MS. But the big downside, and all of that work that I just talked about, was using the original sequence, which took 25 minutes to image one slice. So that's obviously not very clinically useful. Covering the whole brain will take all day. And that's really probably the primary reason it's been pretty restricted in going any further than UBC for quite a long time. But in recent developments, so as part of my PhD, we got up to 7 slices in 20 minutes, which was a good advancement of moving to a 3D technique. And then the next step where we're sort of sitting right now is for people who know about MRI, to combine gradient spin echoes to speed it up a little bit. So we're now covering about 20 slices in about 15 minutes, and this is really exciting because we've got Chase working on this here at your center as well. And then this is some of the work that we're presenting at ISMRM this year, so we're now requiring 20 slices in about 8 minutes, so by making various tweaks to the sequence. So this is exciting, it's getting a lot more clinically feasible at long last. In the meantime, while I was in the UK, I was working with a different technique with all the same principles, but a slightly different acquisition, so I'll just quickly introduce you to that. So it's called McDespot. Stands for multi-component driven equilibrium single pulse observation of T1 and T2. So yeah, it's the same principles. You still have these two different water compartments that have different relaxation characteristics. But instead of doing this fairly simple but long spin echo experiment, we're using steady state imaging. This will be brief for anybody who's interested in MR. So we do a spoiled gradient echo acquisition, and the signal for an SPGR depends on T1 and the flip angle that you apply. So you apply a range of flip angles, and for a given T1 you observe the purple curve. And then we also do some balanced SSFP, where the signal depends on flip angle T1 and T2, and so you observe the purple curve as you vary the flip angle, and then we just fit these two curves to a model that we have these two water flows with each other on T1 and T2, some exchange between them, and then a different proportion. So it's a complex model, so we're fitting 7 parameters, but it's fast, is the really good thing. So it's nice because we can run it on any scanner manufacturer, so it's pretty much ready to run anywhere. The image artifacts aren't really too bad because they're a series of short scans, you don't have to worry so much about things like coughing, which is nice. And it's fast, so we're running whole brain in under 10 minutes at quite nice high resolution. It also works pretty well in spinal cord, so that's a monowater fraction map along the cervical spine in healthy control. And because it's 3D, we can look cross-sectionally, and we were hoping to see these lovely gray matter butterflies straight out of the textbook. And well, it's low resolution, but it's there. I'm fairly convinced that it's lower values in the butterfly, so there's less myelin in the gray matter, so you can get into that. So that's exciting. And it does vary along the spinal cord, so as you go down to the lower spinal cord levels, you get lower values. And this corresponds really nicely to the increased proportion of gray matter there on the C6, C7 level. So again, this is just encouraging that we're measuring something relevant. And it also points out how important it is to get away from that old single slice technique, because if you're just mispositioned a little bit, then you're going to be changing your monowater fraction just from that, and not from any intervention that you might be trying. So the limitations of McDespot is it's a two-pool model. So I talked about, the intra-extracellular water and the myelin water. And for the McDespot, that's all we can fit. So if there's other things going on, we won't know about them. Whereas with the TT distribution, there might be more than 2 peaks and we can characterize them. It's a big, complicated model. We have to make some pretty big assumptions, and it takes about 24 hours ahead to run the analysis. And our values do come out quite a bit higher. Oh, and I should point out I'm using a different abbreviation for the values we get from McDespot, which is fractured of myelin. That's just to differentiate it because it's a different technique. We get out higher values, and we're not completely sure why. We're still working on that. But we do get a really good correlation between what we get with the FM from the McDespot compared to what we get with the sort of gold standard acquisition. So we're encouraged that we are measuring something that's actually at least highly related. So moving on from the development into some applications. So one of the projects that I was involved in Oxford was looking at a cohort of primary progressive MS patients. So in general with MS, most people will present with relapsing remitting MS, which means that they will have an attack. So they might go blind in one eye or get numb, and then about a month later, so they will recover either partially or completely from that attack. And they go through this pattern multiple times. With primary progressive MS, they present as just continuously getting worse. So they don't have these attacks and recoveries. It's just sort of a progression right from the start. And those lesions that I showed you on the conventional image, you don't see that many of them in primary progressive MS, not nearly as many as you see in relapsing remitting. So we're assuming that there must be more damage that's going on in the background, some more diffuse changes that aren't getting picked up in the conventional MRI. And this is quite a critical problem because all of the markers that we use to approve a drug are based on these lesions and on attacks. And so if we don't have those in primary progressive MS, we have no way really of approving a drug at present using the markers that are commonly used in MS. And there is no treatment right now available at all for primary progressive MS. So we need a measurement that has good volumetric coverage, so we can look at whole brain and that, because we don't really know where we're expecting the change. We want high spatial resolution because they're quite small. We want sensitivity, obviously, and specificity so we know what to target, and our best guess was myelin. So McDespot seems like a good fit. So we scanned 17 primary progressive MS patients and 17 healthy matched controls, and we wanted to compare their data and with clinical scores as well. And we thought that we would see primary progressive patients having lower myelin-water fraction than the controls, and that these decreases would hopefully correlate with clinical measures in areas that made sense. So as a first step, shoot, that is not showing the images. That is where. Okay. I have no idea what to do about that because it shows it on my screen. Well, I can probably tell you. Hello. I don't know, do you think it's worth unplugging and plugging back?
Would if you go out of the slideshow?
Yeah.
This happened to me last week and I couldn't figure it out actually. I'd never had that happen before, but just last week this happened to me.
Try one more time. No, okay. This isn't critical, so it's okay. We did find a significant reduction in myelin-water fractions that was spread throughout the white matter for the primary progressive patients, so it was sort of everywhere. And we did, it's okay, there's only one more. We found a significant correlation between the reduction in myelin and the clinical disability. And again, that was sort of spread through the whole brain. But the EDSS score, which we normally use in MS, is not very specific. So we tried to look at the subscores that go into calculating the disability score, and we found that the mental subscore was correlated in sort of the frontal lobes and the corpus callosum, whereas the sensory score was correlated at the back of the brain close to the sensory cortex. So basically these were where we would have guessed that they would be. So that's encouraging. These aren't great scores, but it's encouraging that at least they were in generally the right area. So the next step, and this is something I'm really excited about, is trying to create an atlas of myelin. So if anybody's worked with MRI, they've probably worked with general white matter atlases or DTI atlases. And so we tried to make one with myelin-water fraction. So we took our healthy control population and we figured out what the average myelin-water fraction is at each point in the brain and the standard deviation. So what's the variation at that point across the healthy population. And then you can take your individual patient and compare it to that atlas. So it gets away from the group differences and gives you a tool that possibly a radiologist could use on an individual basis. Okay, so basically what you do is you take your atlas and you take your patient and you just count how many standard deviations away from normal is each point. That's a Z-score. So anything between 2 and -2 should account for about 95% of your data if it's normally distributed. So anything outside of that is relatively abnormal. And if you do a really strict comparison for multiple comparisons, anything outside of 5.4 is highly significantly reduced. So the greens are extremely significantly reduced. So this is a patient with a very low disability score. And basically some of them are just misregistration problems. You can see them right at the borders of the gray matter. But basically you're seeing the green spots where you can see lesions in the conventional image. So a little bit more, so it's probably downstream damage from the lesion, but mostly restricted to the lesions. This patient with a much higher disability score with a very similar clinical image, just a few lesions, you see a lot of green. So pretty much most of their normal appearing tissue is not normal. So it's significantly reduced in myelin that you're just not picking up on the conventional image. Another example, another higher disability, this person did have a lot of lesions, but really they have no normal tissue. Everything was significantly reduced in the myelin-water fraction. So that might be useful as sort of a snapshot, individual kind of approach to this data. So we thought that was encouraging, so we decided to extend it and do slightly better clinical measures and include spinal cord. So we then scanned 15 more primary progressive MS patients and 10 controls. And this time we included spinal cord measurements. And so our hypothesis, so we also looked at atrophy in this study. So one of the more common measures to look at is just has the brain shrunk, has the spinal cord shrunk? And this is a pretty sensitive measure, it's just really unspecific. So we wanted to include that in our study because we can actually calculate it straight off the MacKay spot images, so we had it for free anyway. So we thought we could get more of a complete picture of what's happening by combining these volume measurements with the Myelin-specific measurements. And so we were hoping to basically see the same thing, reductions and correlations where you'd expect them. All right, so these are conventional spinal cord images. So the big black band that you see through, that's because we've actually nulled the signal there, and that's to sort of get away from the effects of swallowing and that, so that's normal. So this is a healthy control and then 2 MS patients, one with a higher disability in the middle and lower disability on the right. And it's a T1 and a T2 weighted image, top and bottom. And I am not a radiologist, but I can't pick out lesions on the conventional images. You can sort of see a hint of one on the bottom middle image, but basically they're pretty hard to pick out in general in the spinal cord. When we looked at the water fraction maps, then it was quite a bit more obvious, so same patients, and so anywhere orange is sort of lower. So you see some orange in healthy control, and that's just gray matter partial volume in the slice. But you see a lot more red in the patients. So we're encouraged that we're probably picking up at least some decreases in myelin in the primary progressive MS. So when we looked at the volume reductions, we did find that the ventricles were significantly bigger in the MS, which is as expected. And we found overall that the gray matter volume was decreased in MS, as was just the peripheral, the cortical gray matter. And the total brain volume was decreased, and the total white matter was not quite significantly, but decreased. So this is all in line with past studies. So this is just showing that we can make the same measurements off of the images we were acquiring anyway, and reproduce that result of the changes in brain volume. Spinal cord was the most significant decrease, so we could pick up a good significant decrease in cervical spinal cord volume as well in the primary progressive patients. And then more interesting to us was looking at the myelin-specific measurements, and again, we reproduced a kind of significant reduction in the white matter of around 15% in the primary progressive MS group compared to controls. And then we looked at a couple of different brain regions a bit more closely, so the minor forceps was quite reduced, and more of a decrease in the corpus callosum. That's the part that connects the two halves. Those of you like me that have no brain anatomy. So that's kind of a nice target to look at, is the corpus callosum. So it's easy to pick out, easy to measure, and it's definitely highly effective in MS. And then the cervical spinal cord, so this is where we found our most significant. There's almost no overlap actually between the monomotor fraction values that we got in MS and controls. So this is a really good place to be looking for a target for clinical trials. And I won't go through this in detail, but the idea is we tried to correlate all of these different measures against the different clinical scores. So we have EDSS, which is just our broad disability measure, which everybody hates because it's way too broad. We have the MSFC, which is a functional composite scale, which is another sort of broad score, but it's made up of the next three tests. So the PSAT is it's supposed to be a cognitive test. It's extremely stressful if anybody's ever had to do it. So I will face you in a small room and read out a list of numbers, and you have to tell me the sum of the previous two numbers that I said, and ignore what you just said, but just keep summing up the last two numbers that I've said. So it's kind of more of an attention and stress test than anything. A 9-hole peg test, which you have a little board with 9 holes and these things that look like golf tees, and you put them all in the holes and then take them back out. And we time that with each hand. So that's sort of a dexterity test, but there's also some corpus callosum involvement because you have to cross the brain. And then a 25-foot time to walk, which is just our ambulation score. And basically we found what we kind of hoped to was that we can find volume measurement changes in the brain with more cognitive scores and myelin changes in the brain with the PEG test and in the spinal cord for the walk. So overall we're describing sort of the different clinical symptoms with this combination of techniques. So then we decided to move on to see if we could tell the difference between two related diseases. Okay, so this study was looking at relapsing remitting MS, which is the main type of MS I was talking about with the attacks and recoveries, and neuromyelitis optica, or NMO. So NMO we used to think was a subtype of MS. It was just particularly aggressive and really went after the spinal cord and optic nerves. But it turns out that there's actually quite a different mechanism. So there's an antibody against a specific water channel in NMO. So it's the aquaporin 4 water channel that is affected. So it's the astrocyte foot process that's actually problem area. So it's a completely different mechanism of demyelination from MS. So same clinical symptoms essentially, just kind of aggressive, different mechanism. And actually the treatment is different. So it's important to get this right. And the antibody test isn't 100% accurate, so there's need for more ways to identify it. And we want to just understand some of the differences in how our imaging methods might pick these up. So hopefully I've already convinced you that in MS, the normal appearing tissue isn't normal, that there's changes happening there. We don't know about NMO, it's pretty rare, and the hypothesis is that it's only really affected in the acute damage, so in the optic nerve, in the spinal cord, and here and there where there's lesions, but we don't know. So this is what we wanted to find out. So we compared 14 NMO, 14 relapsing remitting MS, and 17 controls. And we were kind of hoping and expecting to see a decreased myelomotor fraction in the acute and the diffuse areas with MS, but only in the acute areas for NMO. All right, so this is in the normal appearing white matter. So controls were definitely highest. MS was significantly decreased in the normal appearing white matter, as expected. But NMO, it wasn't significant, but it was more decreased than I was kind of expecting it to be. I was hoping that it would come out sort of equal with the controls. I'll come back to that. We also looked at gray matter, and again, it was much more of a decrease in the MS patients, not so much in NMO, that was variable. And we compared the lesions, and they seem to be about the same, no significant difference between the myelin and the lesions between the two diseases. So let's come back to this went against my hypothesis. So we wanted to look a bit more closely at this, so we looked back at the Z. Okay, you're not going to see that picture. So basically we're trying to see if maybe what's happening is that the patients with higher disability have more pixels that have decreased myelin in MS, but not in NMO. So we tried correlating that. And so the blue dots with the MS, it's not a great correlation, but you can sort of see that the patients with the higher disability have more pixels that were decreased in myelin water fraction. Whereas with the NMO, they're all sort of sitting along the bottom in red, which was basically our hypothesis, except for these 2 points. And these were the 2 points that were driving that bar chart to not look how I expected it. So those are the ones that were showing this sort of decrease in the normal appearing white matter for NMO. So we want to look at those 2 cases more closely. Okay, this one sure. So basically, the NMO patient on the top, so what I've done here is anything in yellow was significantly decreased in myelin, and anything in purple was from the lesion mask. And then there's a conventional image on the right. So patient A actually just had a ton of lesions, and all of the sort of damage to the normal appearing white matter was actually sort of adjacent to the lesions. So it's not like the other MS patients I showed you, where there's damage everywhere, even away from the lesions. So that's probably what's happened. It's just a really quite severe case. The patient on the bottom, we found out after had a biopsy, and so that seems to have been what messed things up. So you can see that there's actually changes in the myelin and lesions showing up all along sort of the affected area from where they went in that biopsy. And that's what was driving that one. So if we take those 2 points out, then we go back and it actually quite nicely fits the hypothesis. So we still would like to reproduce this, and we're working on that now, but we suspect that we were right, that there's a lot less damage in the normal appearing tissue in NMO. In spinal cord, we found reductions in both, so significant reductions in the myelin for both NMO and MS, as expected, as we knew that the aquaportin is heavily involved in the spinal cord. All right, so the last application I want to show you was a disease that isn't traditionally associated with myelin, so this is motor neuron disease. So this is progressive weakness with variable muscle wasting. The most common type of motor neuron disease is ALS, or Lou Gehrig's disease. This is a brutal disease. You have about 2 to 3 years from symptom onset, and that's about it, although it does vary quite a bit between people. A less common subtype is PLS. It's not quite as brutal. You have 10 to 20 years from symptom onset for survival, but it takes about 4 years to get that diagnosis. So essentially, if you outlive your ALS prognosis, then you probably have PLS. So you can imagine that getting an earlier diagnosis would have a pretty significant impact on quality of life. And we're also interested in seeing if there really is differences between these diseases, or if they're just 2 versions of the same thing. We know that there is some demyelination present from postmortem studies, but we don't really know at what stage it comes, because the postmortem samples are generally going to be at end of life. And so it's presumed that this is secondary to axonal damage, but it really hasn't been well studied. So our aim here was to compare. We had 23 patients with ALS and 7 with PLS, that is more rare, and 12 healthy controls. And we thought that we would see a reduction in the myelin fraction in ALS and PLS, and that probably the people with the more severe disease would have the greater myelin loss. Okay, so we found significant reduction in myelin fraction in PLS, so this is the less severe disease, and the smaller group. It was significantly reduced myelin pretty much throughout the brain, so in red was significant after all the fractions from multiple comparison, and blue is just below significance, just to show that we were just below significance pretty much everywhere. So that was kind of exciting, so we weren't really expecting to see anything. But when we looked at ALS, which is the more severe disease, and the bigger group, we found nothing, absolutely no reduction in the myelin water fraction. That's kind of the opposite of what we expected to see. So we thought ALS being the more brutal disease and having the bigger group to get a more significant decrease, but no. So then we compared them directly to each other, and we still found that when we compared PLS to ALS, it was still a significant reduction, just below significance on the other side, so I think it is bilateral, just low numbers for PLS. So we thought, okay, well, this must be an effective disease duration, because pretty much by definition, the PLS patients have a longer disease duration than the ALS. So we tried correlating it with disease duration or age, and there's nothing. We tried picking out the ALS patients with the longest disease duration, which overlapped with the PLS with the shortest, and still it was only the PLS group that had the reduction. And there's no correlation with disability level. So our conclusion from this was that we suggest that it's actually a different mechanism. There's actually demyelination going on in PLS that isn't happening in ALS. So this might actually be something helpful for diagnosis, and we're trying to apply it more prospectively now to see if that's the case. Okay, so I've talked to you a little bit about development of the Myelin water fraction technique and some applications, and hopefully I've convinced you that we can use this to show differences between people with disease and healthy controls, show differences between diseases and each other, and that it's sensitive to some clinical measures. But we still have left to do showing changes, robust changes over time, and probably most importantly, whether treatment helps stop these changes over time. And so that's what we're working on right now. So actually last week, we collected the last data point in this study, which is very exciting. So it's a big clinical trial for a new drug in MS. And it looks really promising. In the phase 2 trials, there were 0 relapses, so it's a really, really promising drug. And we were sponsored to put a big sub-study onto the trial, so we're running a whole hour and a bit of advanced imaging on top of what they're already doing as the main clinical trial, including myelin imaging. And so we're hoping to be able to demonstrate repair and neuroprotection with this drug. And our hypothesis is that this drug will be superior to the sort of basic drug that they give as the other alternative, because it's a blinded trial for relapsing remitting, or because there is no treatment for primary progressive, they're just on placebo, and see how that's affected on our advanced MR measures. And then we're still interested in looking at the correlation between the MR outcomes and clinical scores. And because we had extra time, we put in more techniques, and we're interested in comparing the techniques to each other and combining them. And just out of interest, some of the techniques that we're running, we've got the 2 different approaches to myelin imaging, so the traditional spin echo approach, as well as the McDespot magnetization transfer imaging, which is another sort of more traditional measure of myelin, but it's also really sensitive to inflammation. Susceptibility weighted imaging, where we're getting changes in microstructure quite early. Spectroscopy, which is specific, you're measuring brain metabolite concentrations, but it's also, you can only kind of do one big box, so you don't know where the changes are happening, but really, really specific. And we're doing some rescue state functional MRI to see what the actual changes in function are with all of these diffuse changes. So you'll have to stay tuned to hear the results of that trial. They'll get locked down in a couple of weeks, and then we can look and see if we were able to tell the difference between the 2 groups. And we're really hoping that it will be helpful. They're actually including this data in filing for licensing the drug, which is scary and great. So I just want to wrap up with a couple of trials that we're going on right now. So we have that big trial in MS and another trial in MS. So we have a couple of trials in NMO, which is really exciting because normally pharmaceutical companies are not willing and able to sponsor trials in very rare diseases, but this one has really gained a lot of interest. So it's quite exciting to see so much progress being made so quickly with this disease. We're trying some more applications, so we've been just starting some studies in stroke, Parkinson's, and then also in injury, so traumatic brain injury and spinal cord injury. And I'm really interested in combining some of our myelin imaging metrics with the functional imaging and measures of cognition, which is really understudied in a lot of neurological disease. We still need to understand a bit better how these different parameters relate to each other and how we can best use them. And I think for now, our biggest benchmark is we need to be able to show that they predict whether you're going to do well on treatment and what your likelihood and time course of progression is going to be. And if anybody has any interesting projects, I'm always looking for other applications. So new applications for myelin water imaging or clinical questions we can try to answer. Or also if I can test out your techniques in my applications and basic research and validation. So if anybody has any good ideas for projects, definitely drop me an email. And I'll finish up just acknowledging some of the people I worked with in the UK, as well as the volunteers and their families and our technologists and all of our sources of funding. And finally wrap up with the rapidly growing UBC group. And if anybody is out in Vancouver, we would love to have you come and visit and check out the MR facilities. I'll stop there. Thanks.
All right, Zara.
Hey, thank you. Nice talk.
Thanks.
Question. Recently, quite recently, actually, just last week or last 2 weeks, I saw a paper published talking about existence of different myelination in the brain. And it was claiming that the signal transmission, therefore through different regions that have different myelins, is quite governed by the different mechanism. I wonder if your technique can distinguish different myelination and whether that reduction has to do with the difference in myelins.
That's really interesting, yeah. We definitely do measure quite different values in different brain regions, which is one of the things that we think helps validate that what we're doing is related to myelin, because we do know that there's different content. I know that there's different composition in the spinal cord than there is in the brain, but I'm not a biologist, so I don't understand the differences in different regions probably. Other people in the room could answer this question better than me. We did find that, for instance, we were interested in the corticospinal tract because that's an easy target to sort of look at functional change with. And when we looked at it, it looked like it had a really high myelin water fraction. But we looked at the distribution of the T2s, and it turned out that it was more that the peak from the intercellular water was a lot broader there. And so it was actually bleeding into where we cut off for what we think is myelin. So it's actually something to do with the biology of the cells in that area. So maybe not about the myelin in particular, but definitely about how the cells work together. So that's a very long answer too. I think if we look at the whole T2 distribution, we can definitely get information about different composition potentially or different function of myelin.
Just as sort of a follow-up to that, I know like Sean has been doing some work on development, quite a lot of work it seems like actually. I wonder if there's anything to be gleaned from that in terms of the development of, myelination in different regions over the course of, maturation during childhood.
Yes.
Do you know what they found with that? I haven't really kept up with that.
Yeah, no, I mean, so they've been scanning babies basically, which is a lot of work. It means you stay up all night, wait for them to fall asleep, and then try to keep them asleep while they're in the scan.
And that's after fighting with the ethics people.
Exactly, yeah. It's more fighting with getting people to sign up, because if you're trying to do DNA sampling, you get really worried about it. Anyway, so they have been able to show the development of myelin over with age, and it basically followed more or less what was expected from pathological studies, with a few minor differences. So some areas that seem to develop more quickly than expected, and I think there was sort of a gender bias earlier on than they expected as well. But I just think it all fits. The areas that are the last to develop are the most vulnerable, and those seem to be the areas that are kind of, I wouldn't say they're necessarily the most hit, but probably when they are hit in disease, they're the less likely to recover. So there might be something there. And then also, I was talking about this complex model that we have, right? So we have the T1 and T2 of the myelin water, we have the T1 and T2 of the intercellular water, we have the exchange, and then we have the proportion. So if we look at some of these other parameters, instead of only ever looking at the myelin water fraction, that should tell us more about it. So for instance, exchange is telling us how long it takes essentially the water to exchange magnetization, so either to escape from the myelin sheets and get out, or to sort of come in contact in some way. So if myelin is thinner, we would expect a faster exchange time, for instance. So we can start to look at things like that. And we do find a longer exchange time in spinal cord where there's thicker myelin. So probably we can tease something out with the rate model.
Questions, Ken?
I have a question on the slide where you had the Z-score and the color map for that. You had mentioned something about multiple comparisons. I'm wondering, what were the multiple comparisons? I might have missed it.
No, I didn't explain it. So basically, if you're really strict, basically, whenever you compare more than once, then you increase the likelihood that it was a chance finding. So usually we use a P of 0.05 to say that something's significant. So if you do multiple tests, then you have to divide your 0.05 by however many tests you do, and that's what level you have to reach to be significant. So that's doing a strict comparison for every voxel in the brain.
Oh, I see. It was overall the voxel.
Exactly.
Okay, I understand.
Yeah.
Thanks.
So I think most people would say 3 or 4 is probably a reasonable place to look, but if you're really, really strict, then it comes out to 5.4 for this size of the data set.
So that's just comparing one brain?
Yes, one brain to the atlas.
To the average, or just one?
To the average.
To the average of everybody in your control?
Yeah.
Okay.
Yeah, so in this case, and this is something that I'm really interested in doing, so in this case we just had the controls from this study, and so they were sort of age and gender matched to the group. But what I'd really like to do is do a big multi-center trial, so we get data from lots of different places, huge age range, and then get as much information as we can about the controls so that when you have a patient, you can select and make an atlas of just females aged 20 to 25 non-smokers kind of thing, so that you can really get sensitive to changes from what's expected. That's the goal.
Paul?
I've got a question. So when you're comparing these two images together, how do you account for the fact that the different brains will have slightly different sizes?
Yeah, no, that's an extremely important question, and some people dedicate their whole career basically to that. So trying to register images into some standard space and to each other, and it's especially a big issue in MS because these ventricles, these areas in the middle, get bigger as the brain shrinks, and so you can actually see these ones look kind of weird and distorted, and that's because they've been warped to a standard space. And so it's not a simple answer. It's something that people work very hard on, of trying to decide how to best match these up, but there's algorithms that look for differences in intensity or for edges or things like that to try to match them up quite carefully. I've been working a lot with a guy who used to work with Sean as well, and they were working with babies, and so the brain is not only changing size rapidly, but actually the contrast changes as the development happens. So trying to make templates to register to in that space is even harder. So he thought MS was easy. So yeah, it's an important point, and one of the difficulties of doing this atlas approach is trying to do that well.
So in some of the places that were highlighted, I think in the subject before this, it's there, where it's right around the ventricles, like in the top.
Yeah, so you sort of see like these spots here, that's probably just because it's badly registered.
Gotcha. Yeah, I was wondering,
Whereas
like...
this is real, and this here is real.
Sure. Were you using some sort of high-dimensional nonlinear warping?
I was at FIMRIB at the time that I was doing
Okay.
this, so yeah, we did a 5-step nonlinear
Right.
registration
The
that gradually
nerd.
went, yeah.
Yeah, okay.
So, yeah.
Gotcha.
Is diffusion tensor imaging a broaden approach to this problem? And if it is, did you compare the results?
Mm-hmm. Yeah, that was actually a big part of my PhD thesis. So diffusion tensor imaging is extremely useful. So basically what that is you're looking at the movement of water, and it preferentially will move along a myelinated axon rather than across the myelin. And so you wait some time and see which way the water's moved, essentially. So that's great because we measure how much more likely is it to move in one direction than other directions, and that number will be higher when you have myelin. So when myelin gets hurt, then this number goes down. The problem is that it also is affected by axonal membrane loss, increases in water, gliosis, all these things have a pretty big effect on diffusion. So you have to be really careful with the interpretation. And the other problem is that when you have areas where the fibers cross, then you get sort of nonsense numbers there, which we don't have a problem with the mile-water fraction. So we tried correlating sort of the FA with mile-water fraction, and it works pretty well within white matter structures, but you sort of see them grouping here, there, and everywhere on the graph. And it's just probably because you have certain areas that will have a pretty high myelin content, but maybe are less cohesive. Like the corpus callosum is really nicely all oriented in one direction. So you get a really high FA even though the mile-water fraction isn't that high there, and so on like that. And then in disease it kind of all falls apart. It works really well for acute injury models. So like if you have spinal cord injury or something like that, then it works quite well. But in these messy cases where you have all these different things going on at once, it's just harder to interpret. So we generally tend to also include it in the studies because it's really useful for finding out where the different tracts are. But we have a hard time with it in MS because the lesions sort of mess that up as well. So yeah, definitely useful, but we think this is probably more specific, best used in combination if possible.
Any other questions? Okay, I'll ask one more that I was kind of curious about. Because you just touched on this, I think, in one of the box and whisker plots, but you had mentioned about gray matter differences, actually.
Yeah.
And so I was curious, whether you guys have noticed any differences in myelination in gray matter. It's something, again, that you could not do with DTI, really.
Yeah.
Because the FA is sort of not going to be there anyway. But yeah, whether I've heard a lot more lately about gray matter lesions in MS, and this is, I think, a growing topic. Have you guys noticed any changes in gray matter?
It's brand new for us to look at it. So with the traditional technique, we didn't have enough signal to noise to pick anything up in gray matter. With the latest and greatest sequence, we're doing okay if we take an exon acquisition. With the McDuff spot, you do get signal in gray matter. And so that's where I was sort of starting to look at it. It doesn't help us to pick out gray matter lesions per se, but I think that's mainly a resolution issue because they tend to be quite small and they tend to hug sort of the boundary between the gray and the white matter. But it's definitely something that we want to look more at. And so we have a student right now who's just developed another new way to segment cortex that seems to work really well in MS. And so using that, we're hoping to get a more accurate delineation of what we're looking at.
Sure.
So it's another one of these things where you have to be used to looking at these images to look for abnormalities. And it takes years of training for radiologists to be able to pick out abnormalities, and so to train them on a whole other image is non-trivial, and I'm sure not going to pick it up. So we need mathematical ways of doing it, and that's still up in the air.
Sure.
Yeah. So yeah, I would definitely like to look at that more and tie it in with some of the cognitive measures and functional.
Yeah, exactly. That's what I was wondering, is if you could maybe tease this out. I think that would be another very clear advantage of this method. If you could actually detect things like that, that would be huge.
So my student who's defending tomorrow, she's been looking specifically at the visual pathway in MS and NMO. So she's looked at people whether or not they've had optic neuritis, which is a lesion on the optic nerve. And then we did OCT, which is measuring retinal thicknesses, essentially. So you can see sort of the damage going that direction. And then we did myelin water fraction in the optic radiations, so what's happened to the myelin downstream. And then also in the sort of visual cortex, we tried to look at the myelin values there as well as the thickness. And it looks like in MS the damage is pretty independent of the optic neuritis, although it's worse when there's optic neuritis. Whereas again with NMO, it's optic neuritis driven. So it's sort of another model that we're trying to look at there. It's a nice system.
Yeah. Okay, if there's no other questions, please join me in thanking Shannon again for this talk.